All right, so here we are. We are still in chapter 9.1. This is going to be part 2. So we're going to finish up the chapter talking about first our alternative patterns of cell division. So recall that at the beginning of 9.1 we talked about binary fission, and binary fission is our most common type of cell division that we see in prokaryotes, but we do see a couple of other ones. We're going to talk about 2 more specifically, and then we're going to spend some time talking about biofilms. So first, our alternative patterns of cell division. And so again, remember, binary fission is our most common pattern, and we see this really in most things. There are just kind of a handful or so of different other types. The first one is fragmentation. And in fragmentation, this is when we have within one cell, we're going to have many nucleoid regions are going to form. And then kind of whole cells in themselves will be forming. And then what they do is they split away. So we'll have many of them kind of just break off and kind of float away from this kind of parent cell as these cells have been formed. This is a process in which many new cells split away from a parent filament. So we see this in, for example, actinomyces. In actinomyces, these are anaerobic soil organisms. Soil-based, I guess. They live in the soil. And then also we see this in some cyanobacteria. So the way this kind of looks, I'll use green because we see this in cyanobacteria, but so kind of we have this filamentous cell here that kind of forms these other nucleoid regions or these other cells inside of it. And then what happens is these cells are just going to break away and fragment off and float away. So we have our original cell up here that is just kind of elongating and creating more cells within it. And then at some point these cells are going to break off or fragment, break away from the parent cell. The second one that you should know or be aware of is called budding. So in budding, this is where we have a reproductive division, but it's unequal. And so then this smaller cell is going to detach from a parent cell. So it's an unequal. So if we think about binary fission, remember in binary fission we had the one big cell that then, split into 2, and then we had, the 2 cells that were pretty much equal in size. With budding, we have an unequal reproduction division. Or reproductive division, where the smaller cell detaches from the parent cell. So in this case, we'll have a rather large parent cell, and then it'll start to kind of form this cell, this additional piece of it, kind of off of the side of itself. And then as it does this, it's going to start to put these things into the cell. So all of the different organelles and things, not really organelles, I apologize, but all of the various things that a cell will need, depending on the type of cell. And then even the nucleoid region will form. And then what will happen is we have this kind of breaking off, that's not quite cytokinesis, but a breaking off of this cell. And then this will be separate. So again, it's uneven though. So we don't have this kind of in binary fission, this very equal separation and division. In this case, it's kind of building up this additional smaller cell, and then that eventually breaks off. And some of the difference here, or at least one difference that you should be aware of between fragmentation and budding then, is that in fragmentation it kind of forms several of these nucleoid regions, therefore several of these different cells, and then they kind of just break away and disperse, several of them at a time. Versus with budding, it's that cell is kind of putting all of its reproductive energy into that one cell, and then that one single additional smaller cell breaks off. All right, so now let's take a look at biofilms. Now, biofilms are a very important aspect to studying microbiology because biofilms are very important in human health. And not even just human health. We see biofilms in myriad places. So we see them, in the bodies as far as how it's related to human health, as I mentioned. But then also we see them in lakes, we see them in streams, we see them, floating in oceans. We can see them form in our pools and ponds and things like that. So we see biofilms in many places, and they can be very detrimental. So what is a biofilm? Let's start with that. So biofilm are colonies or ecosystems, really, because it can be many different types of microbes, and we'll talk about that. But of microbes that form on a surface or interface. So in this case, they're communities, and biofilms are very highly structured. So let's write that down. So communities are highly structured and provide advantage to constituent microbes. So rather than being alone. Okay. So when we're talking about biofilms, what we're talking about are various different types of microbes all kind of living together in this area where they actually create what's called an extracellular matrix, which we'll talk about, around themselves, and then they communicate with each other. So that's where the highly structured comes in, at least partially, is that they create a structure around them for all of these different types of bacteria and other microorganisms to live within. That's going to provide an advantage or be beneficial to all of the different organisms within that community. And then also it's going to prevent them from being destroyed by antibiotics and other disinfectants and things. And that's going to be much better for all of the different organisms compared to being on their own or being alone. So they'll form these and then they communicate within these communities. And then we have a biofilm, which can be very difficult for us to get rid of. So let's take a closer look at biofilm structure. So the first piece are called streamers. And we can see streamers in fast-flowing water. So there are kind of two ways to look at biofilms. We can see biofilms in slower-moving water or slower-moving fluid environments, and then we can see them in faster-moving fluid environments. So streamers are going to be in the fast-flowing. So fast-flowing water. So in this case, what we're talking about is there's actually an anchor. So they're anchored by what's called a head. And then the tail portion is going to flow in the current. So we'll have something that's kind of anchored up here, and then we have kind of the tail portion that's going to be flowing in the water, which is fast moving, but it'll be kind of hanging on, and we'll have maybe various types of streamers if this is upstream, for example. So this head portion is maybe attached to a stone, and then the rest is flowing down in the water. So this is in fast-moving water where we have this anchor and then this flow. When we're talking about slow-moving water, we have a typical mushroom-type shape. So this is in slow-moving water. And then in this case, even though it mainly adapts this mushroom type shape, the shape can also change with different environmental conditions. So the shape can change with different environmental conditions. So if things become more dry, it might change the shape based on that. If things become more wet or more fast moving, then it might change the shape depending on that as well. So within these structures, whether it be streamers or the mushroom shape or any other different type of shape, these cells will come together, and we'll talk about how they're formed. But what we find in the biofilm is that we have this population of microorganisms. But within this population, or in addition to this population, we have an extracellular matrix. And this extracellular matrix, extra meaning outside of, and then cell, so outside of the cell, and it's a matrix, so that's this kind of mesh or netting. This extracellular matrix, we see this in lots of different types of cells. In biofilms specifically, they're called extracellular polymeric substances. So what is making up the extracellular matrix in these biofilms are called extracellular polymeric substances, or EPS. And so this is actually secreted by the microbes that are in the biofilm. So secreted by the microbes within the biofilm. And oftentimes what the EPS is made up of are polysaccharides. So made of polysaccharides. So we have polysaccharides that are extruded or secreted by these microbes, and then it forms a matrix. So all these polysaccharides, so polysaccharides, poly meaning many, and then saccharides meaning sugars. So these long chains of sugars are going to be secreted by these microbes, and they'll create a kind of netting. And what this ends up looking like and being like is a gel matrix. So it's this gel matrix that contains these polysaccharides, but then it can also contain different proteins to give it structure and strength. It can also include nucleic acids within there as well. And so this is a gel matrix, and as we think of a gel, it is going to make sure that we have lots of hydration. So it's a very thick, wet gel substance that has some proteins and nucleic acids in it as well. And then all of the microbes are going to be kind of embedded or a part of having this around them, the extracellular polymeric substances. So when we talk about the EPS, it actually makes up about 50 to 90% of a biofilm's dry mass. And this is important because what we're talking about here is at least half, and most times more than half, of a biofilm is made up of all of these extra things that are going to just be helping and supporting all of the microbes within the biofilm. So it can be very, very thick, not something that we can see with our typical bright field microscope. We need to have something like a confocal microscope, for example, in order to get through these big, thick layers. The properties of these are varied. Properties are varied. So when we're talking about these proteins, these nucleic acids, we're talking about the polysaccharides, we can see different proportions of the proteins versus the polysaccharides, not only based on the microorganisms that we find within this EPS in the biofilm, but then also the environmental conditions. Due to the microbes, microbes and or environmental conditions. So when they form this EPS, they actually have channels within this matrix. And then these channels are actually going to allow for movement through them. So these channels allow for movement of things like water through the EPS. It's going to move things out like waste and then gases as well. So gases oftentimes are byproducts. So we'll get rid of the waste, we'll get rid of gases, we'll bring in the water, and then that's because the water is going to also perhaps contain the nutrients. So they'll form with these channels that allow the movement of these things, which is going to hydrate and nourish and remove the waste for all of these different organisms. So it also is going to protect and shelter, or protect or shelter these microorganisms, microbes, from predators. So predators meaning other microbes, but then also meaning something like antibiotics or disinfectants. So really this EPS is important, and that's what makes biofilm so difficult to get rid of, is because they create this big, thick, gel, sticky, polysaccharide layer with proteins and nucleic acids and things that have channels that allow water to go through and allowing the nutrients to go through and the waste to get lost, and provide protection through this big gel matrix from predators, things that might interact poorly, when we have those different communities that don't act very synergistically, but they're going to be acting competitively. But then antibiotics and disinfectants have a really difficult time getting rid of biofilms as well. So let's take a look at the formation of biofilms. Formation of biofilms. So first, let's do a couple of definitions here. So first is planktonic cells. So planktonic cells are free-floating cells. So free-floating microbes that live in an aquatic environment. So these are the cells that are just kind of out there on their own. They're floating, going with the flow in either fast-moving or slow-moving water, and they are on their own, living in the aquatic environment. Then we have our sessile cells, and these are cells that are attached to a surface. So again, not free-floating, but maybe at this point on their own. Or in groups. So we have our planktonic cells and we have our sessile cells. So when we take a look at the formation of biofilms, we have our various steps. So the first step is that we have attachment of the planktonic cells to some sort of surface. And this surface is generally coated with some sort of conditioning film. This can be something like a slime from some other organism or an algae kind of base, something there that's going to allow the cell to attach to it. So in step one, we have attachment of planktonic cells to a surface with a conditioning film of organic material. So that is step one. Within step one, so once this has attached, now this of course is called a sessile cell or sessile cells because we've gone from planktonic to attaching to a surface. So now these sessile cells are going to start expressing a different type of phenotype. So these sessile cells start expressing a new phenotype, and this phenotype is what is now going to facilitate the formation of this EPS matrix. So sessile cells start expressing phenotype that facilitates the formation of the EPS. So now we have these cells that are starting to express this new type of phenotype because they're no longer planktonic and floating around. Now they're stationary and they start to form the EPS. So in stage 2, now these cells are irreversibly attached. And I should have mentioned that up here in step number 1, these are now reversibly attached. So this is reversible in step 1. So as soon as they attach to the surface, these sessile cells, at this point it's still reversible. They can get brushed off or broken off or eaten off or degraded or any of those types of things. Now, when the sessile cells start to express this new phenotype where they start to produce the EPS, now they have started this EPS, which is this gel-like matrix that then now have them being irreversibly attached. So they no longer can just be brushed off. In step 3, then, we have the biofilm developing. And what that means is that at this point we have lots of growth of the cells and development. And this development, what I mean is development of the EPS. So what I mean by that is that we start to have these different structures. So these channels are beginning to form. They're adjusting and making things like fimbria and pili and flagella so that they interact with the EPS. So they're developing these different types of living, growing and then developing these different types of living by creating this EPS. And then I'll say something like creating channels, for example, which we also see in step 4. In stage 4, this is where we're going to also see things like where they're creating channels and then they're creating this extended matrix. And we have the addition of lots of other cells. So we have water channels, we have an adjustment of the EPS, so changing the composition if needed. And then in step 5, we see that the outer cells are going to now revert to planktonic cells, and then they start to disperse so that they can go elsewhere and become sessile and create this EPS and create an additional biofilm. So the outer cells revert to planktonic and disperse. And then some secondary colonizers attach. So if we take a look at this whole process, it really depends on the type of organisms that we're talking about here. Of course, step 1, just the attachment of these planktonic cells is just going to take place within seconds. So this is just a matter of seconds of attaching these planktonic cells that get stuck, and then they start to create this EPS. Once they start to create the EPS, then they are considered sessile cells. In the second step, then we're talking about them being irreversibly attached because they now have this EPS surrounding them. And so them becoming irreversibly attached and then creating this EPS is, again, this is within seconds because they're starting to change their phenotype to minutes. So they have to change their phenotype and start producing and expressing this new phenotype and in creating this formation of the EPS. Then as this biofilm is developing, so as we have growth and we have cell division here, so cell division, this is taking place within hours or days, depending on the type of organism. So within minutes, we have these sessile cells. Now they're stuck, irreversible, and then they're growing and developing and adding new things to them. Then in step 4, where we're starting to really create these channels, I would say this is mostly in step 4 here, where we're really creating these channels and we're extending out the matrix. This again is going to be within hours and days, depending on the organism. And then when we get to step 5, where we start to have these planktonic cells that are dispersing from the surface, this is really going to be after several days or even months. And then they're going to be spreading, basically. So your text has an image here, but if we say that we have the surface here, then we have our first cells are going to be kind of floating along with their flagella, for example, and then they become stuck here. So this is our surface, and they become stuck. Then they're going to, again, change their phenotype. Let's pretend that's going to make them red. That's our new changed phenotype. Is now we have this changed phenotype into red cells that now are starting to produce this EPS. Then these red cells are going to divide. So we have our phenotypically changed cells, they're dividing, and they're also producing this EPS. Then these cells are going to add more complex structures to their EPS. So now what they have, I'm just going to draw single cells. Oh, because they lost their flagella, of course. So I take this back. So these shouldn't have flagella. I apologize. Because they are sessile. So they no longer need a flagellum because they're no longer moving around. So they are sessile at this point. They don't need the flagellum. And now they are individual cells, but now as individual cells, they are producing these kind of mushroom type structures, for example. And these are all individual cells that I'm drawing here. And then in between these individual cells, we have the EPS. And you can see that I've kind of drawn in some channels here, or a channel, where we could see water flow between here, which will then allow nutrients to be brought to the cells and allow wastes to be removed from the cells. Then as we move on to the next step, we have our, well, here, we have this, right? But now the top is going to kind of burst open. This extracellular matrix, this EPS, is now going to be broken open a little bit, and now we have these cells are going to revert back to planktonic cells with their flagella, and now they're going to move on to the next location. And so that is kind of the structure or the formation of a biofilm. And then we start over again, right? Because then these cells are going to be the initial cells here that started out as planktonic cells, but then became attached. And after they became attached, at first it was reversibly attached, but then they changed their phenotype and started to produce this EPS. And once they produce the EPS, then they are now sessile. And I can probably write that here. So these are planktonic cells. We'll just keep these black here. These planktonic cells, because they have the flagella, they've been moving around and then they get stuck on here. And then they they change their phenotype and they make the EPS. And now they are sessile cells. Now in a biofilm, they have gotten very, very smart, right? So the biofilm is there to help microorganisms. Now what we see is they can actually become very highly developed in this way. So for example, one microbe's waste is then going to become another microbe's food, for example. Or on the surface there might be aerobic organisms or aerobes which consume oxygen, utilize oxygen for their metabolic processes, but then below that we would have our anaerobic organisms. So the aerobic organisms, the ones that can be exposed to oxygen, are going to help to kind of protect or cover up the anaerobic organisms underneath. So they can develop these relationships. So develop relationships within the biofilm. So for example, one microbe's waste becomes another's food, or aerobic organisms are going to help protect anaerobic organisms. So how do they do this? How do biofilms develop these relationships? And when do they become bothersome or virulent to an organism if they are a harmful biofilm, for example? Well, how they do this is through quorum sensing. And quorum sensing is a specific mechanism that is going to allow cells in a biofilm to coordinate their activities. So they can use these little molecules called autoinducers to communicate with different species and with their own species. So they can communicate with other organisms, and then they can all adjust or change or coordinate their activities based on what's best for the group and based on environmental conditions. So the mechanism by which cells in a biofilm coordinate their activities in response to environmental stimuli. So this can occur within a species and between different species. So they can communicate within their own species, and they'll use a certain type of molecule for that, and then they'll communicate between different species and use a certain type of molecule for that. So they can talk amongst themselves and talk in the entire community. And then as they do this, as they communicate, again, they can coordinate their activities. So depending on the environment and the changes in the environment, or based on the type of organisms, how they're going to work together. These molecules are called autoinducers. So autoinducers are small, diffusible molecules that signal, which means that they are released and bind. And what they do is they signal the cell density of the biofilm. So these autoinducers, again, small molecules, they can send these small molecules out, and the more of these small molecules that bind, the more they know how many of them there are. So the more molecules there are, the more of them there are. And so then they can keep sending these signals out and sending these signals out, and it kind of gives them an idea of the cell density. Now, when this reaches a quorum, so when it reaches a quorum, so what we could also call a critical threshold. A critical threshold. The autoinducers are going to signal for the production of different molecules or functions that are beneficial at that density. So when it reaches this quorum, which is some critical threshold, now these autoinducers are going to signal for the production of molecules or functions that are only beneficial at those densities. So what that means, for example, when we have a quorum or a critical threshold, then these molecules being sent out, these autoinducers, are going to send the signal saying we're at this number, basically, of organisms within this biofilm, and then everybody can now turn on their virulence factors. So express their virulence factors. And that may be because they want to wait until they get to a certain threshold to where when all of the organisms within the biofilm start to express their virulence factors, they're pretty confident they're going to be able to take over the immune system or take over the organism. Basically, they can overcome the host. So if there are only a couple of dozen of cells, at that time they're not going to waste their energy and their, base molecules on producing virulence factors, because there's no way that they're going to be able to beat the system or beat the host. So they will wait and they will form these biofilms, and they'll get to a certain level, this critical threshold, before they then start to release their virulence factors. Once they release their virulence factors, that's because they have a pretty good shot at continuing to exist. Now, these autoinducers are a little bit different depending on the type of organism. So we have our N-acetylated homoserine lactones. And these are used by gram-negative organisms or gram-negative bacteria. And then secondly, we have our small peptides. And these are used by gram-positive organisms or bacteria. So this quorum sensing is going to use these two main molecules. So we have our gram-negative bacteria that are going to send out their autoinducers. We have gram-positive bacteria that send out their autoinducers. And then again, this autoinducer is going to bind when this threshold density is reached. So autoinducers bind, and then when the threshold is reached, then there's a cascade of events. So cascade of events, meaning that there's a change in gene expression. And that's because of all of these autoinducers binding. It says, okay, now is the time, and then we see this gene expression change. So when we talk about it as far as human health, I've kind of been mentioning it that way. But biofilms in, not all biofilms are bad in human health. So for example, we have biofilms in our intestines. And these biofilms in our intestines are good, right? Because they're going to help break down food, for example. They help us break down food. We have this just layer in all of our mucosal folds in our intestines that are just coated in our brush border, as it's called. All of these bacteria are in there, and they're creating this biofilm. Or at least we hope they are, because if they are, then we're going to be healthier. Because they're in our intestines, they help to break down our food, they help keep our normal digestion and metabolism happening. We also see some helpful biofilms in our respiratory tract. And these biofilms are going to help keep our respiratory system clean, right? So it's going to help to have competition. So if we have a whole bunch of biofilms, and we see this in the intestines as well. If we have a whole bunch of bacteria that are healthy and happy and not pathogenic, not causing disease, then if we happen to breathe in some particular pathogen, all of the bacteria are going to be so happy and healthy within our respiratory system and utilizing all of the nutrients that there isn't going to be any space for that pathogen that came in. That pathogen is going to need nutrients, it's going to need water, it's going to need a way to get rid of its waste and get rid of gases, for example. And if there's this big, thick layer of bacteria, so if I pretend that this is either the intestines, which of course would be something more like this. But we're going to pretend for right now that this would be like some sort of tube, right? So either the intestines or a bronchiole in the respiratory system. So we're going to say that, actually we're going to say this part here. We're going to remove this bottom part. So this is our tube up here. So in our tube, if we have bacteria that are coating the entire inside of our tube, whether it's intestines or respiratory tract, you can see that if it's completely coated, if we have a pathogenic bacteria come in, then it's going to have a really hard time getting in here and trying to get any access to any nutrients. So if we think about this EPS now being a part of this, if we have a good, healthy microbiome in our intestines, then we have a good, healthy EPS. We have all of these bacteria in here, and then this pathogen here can't get any of the nutrients. It can't get into any of the stuff. It can't make its way through these cells and get into the body at all. So that's why it's important to have healthy intestines. It's important to eat healthy probiotics and prebiotics to make sure that we're maintaining a healthy microbiome in our intestines. And we see the same thing in the respiratory tract. So if we have a whole layer of bacteria in the respiratory tract, if we breathe in this pathogen here, it's going to get stuck here and it's just going to get destroyed because it can't get to the nutrients. It can't get to the water source. It can't move through either and get into the rest of the body because we have this healthy layer of microorganisms, this biofilm. Now, then, of course, we have the opposite, which is what we're often talking about with biofilms, like, for example, dental plaque. So dental plaque is a biofilm. So it's a biofilm that is very, very sticky. So that's why it's plaque. It gets stuck on the teeth, and that has the bacteria in there that are hanging out and can be eating away at the teeth and getting nutrients that way. And then, of course, in that last stage, we have more planktonic cells that are going to be released, move through the fluid in our mouth, our saliva, and attach onto another tooth, for example. Also, we see some in wounds or infections. We can create biofilms. Where we often see biofilms are in medical devices. So medical devices meaning on catheters, which is really dangerous. So on catheters, on contact lenses, on implants. So we often see these in something like fake heart valves. So when a heart valve needs to be replaced, we can see a lot of times we see biofilms build up on heart valves or in artificial joints. So this is when we have a problem with biofilms, is when they are going to start to build up in places like, for example, in the heart valves, we don't want a giant biofilm in our heart. But they are very difficult to get rid of. So if we have a pathogenic biofilm, then why is it that it's so hard to get rid of them? So pathogens and biofilms are more resistant to antibiotics and disinfectants. Now, why is this? Well, for one, the deep cells in a biofilm are metabolically inactive. So, metabolically inactive. So, what that means is they are not metabolizing, right? So they're just hanging out. They're kind of in a holding pattern. And that's because they are kind of stuck in this matrix. They're not needing for anything. They're hydrated. They're just hanging out. However, if the environment changes, then they become metabolically active again. But while they're not metabolically active, then our antibiotics are not going to kill them. because our antibiotics are designed to go after and change or stop the way that they are going to reproduce. So if a particular antibiotic, for example, is going to stop the formation of their cell wall, if there is a cell that is not metabolically active, it's not actually trying to make cell walls or go through binary fission, then an antibiotic isn't going to do anything to that bacteria. It'll just move right along and not do a thing at all. So that's one reason why it's very difficult to get rid of biofilms, is because we just have some cells that are hanging out. And then even if antibiotics are going to work and get rid of those metabolically active cells that are more on the surface, then once those metabolically inactive cells are exposed, over time they can change into metabolically active cells, or they'll just stay metabolically inactive and they'll just be stuck in this EPS and stuck in this layer. Until other cells come along and interact, so the antibiotics will just move right past them. Another reason is this EPS may slow the harming agents from reaching very deeply in the biofilm. So the EPS may slow harming agents from reaching deeply. Also, phenotypic changes. So not only will the EPS just create this big, thick gel, so the antibiotics just can't get in there and get past this big, thick gel, but then also phenotypic changes, meaning that the change that because the biofilm has formed and they have this quorum sensing where they're kind of talking to each other, they can talk to each other and say things like, hey, why don't you increase your efflux pumps? And then what that means is that when an antibiotic comes along, it can pump out the antibiotic. It can, once the antibiotic gets into the cells, the cell will have a pump already in place to pump it out so that it doesn't get harmed. So these are phenotypic changes. So for example, biofilm cells increase these efflux pumps. And then these efflux pumps are going to pump out antibiotics. So again, part of this quorum sensing, they say, hey, there's enough of us here that we are now all going to switch and we're going to start making the genes for these efflux pumps, and we can pump these antibiotics out. Also, this is an ideal environment for horizontal exchange of nucleic acids, meaning that we can move extra chromosomal DNA from one bacteria to another bacteria, one organism to another organism. And oftentimes what we find on these extracellular chromosomal DNA pieces called plasmids are antibiotic resistance. So even if a particular organism or species altogether within somebody's body didn't already have antibiotic resistance, if it gets stuck within this biofilm and starts exchanging genetic information with other cells, those cells may be resistant to an antibiotic. So if those cells are and they exchange that particular DNA, then all of those cells are going to now become resistant to that antibiotic. So an ideal environment for exchange of extra chromosomal DNA, meaning plasmids, for example, or other extra chromosomal DNA. So just chunks of DNA that are kind of hanging out, even if they're not considered plasmids at that point, and they just have somehow been brought into the cell or picked up in the environment. And oftentimes these have antibiotic resistance on them. All right, so that wraps up our 9.1 section.